So what we're going to be talking about first is how the receptor tyrosine kinase will go through a whole process and mechanisms to end up phosphorylating MAPK. So first thing is I drew a cell membrane, and inside of it is the receptor tyrosine kinase. Here's the receptors, and what's going to happen first is a ligand or some sort of outside substance or chemical will go and bind to one of the receptors. After the two ligands or one ligand binds to the receptor tyrosine complex, the two complexes over here will then converge together and meet in the middle, and the parts coming down below will then cause something called autophosphorylation, which will add phosphate groups to the ends of each one of the buds. After the autophosphorylation of the receptor tyrosine complex, it's going to activate GRB2 and SOS, which then SOS will then travel across and then activate the RAS protein, which will take a GDP, remove it, and then add a GTP to cause GTP activation of the RAS. After the phosphorylation of RAF, it then will end up phosphorylating another protein that is called MEK. This will start the whole process of the phosphorylation cascade. MEK will then become phosphorylated again to have two phosphate groups on it. And then from that, MEK will then go to MAPK, or MAPK, which will then phosphorylate that. This MAPK with phosphorylation will then be phosphorylated one more time to end up having two phosphorylations, which will then create a transcription factor that can enter the nuclear envelope of the cell. The overall concept that is occurring here is the receptor tyrosine kinase will have a ligand bind to it. After this happens, the process of autophosphorylation will happen and the activation of GRB2 and SOS will occur. From there, the RAS protein will become activated by GTP. RAS that is activated by GTP will then activate the RAF and phosphorylate it, which will then create a phosphorylation cascade. First starting with MEK with two phosphorylation or phosphate groups, MAPK, and then MAPK will become phosphorylated to cause a transcription factor, which will be able to enter the nuclear envelope and then act upon the DNA.
Now we'll be talking about the G protein link receptors and how it creates the secondary messengers for CAMP and calcium. So here we have inactive trimer, G protein, which then gets activated by a signal from the ligand, which then binds to the receptor. After the signaling from the ligand to the receptor, it activates the G protein, which has a GDP here, and after that it separates the alpha from the beta gamma, and also, while it does that, it removes the GDP and gives it a GTP. After the removal of the GDP in return for GDP, the alpha then binds to adenylyl cyclase and activates it. After activation of adenylyl cyclase, it uses ATP and converts it into cyclic AMP, which then converts that into protein kinase A. After the removal of the GDP in return for GDP, the alpha then binds to adenylyl cyclase and activates it. After activation of adenylyl cyclase, it uses ATP and converts it into cyclic AMP, which then converts that into protein kinase A. Another thing the alpha G protein can do is activate phospholipase C. So after activation of phospholipase C, it cleaves PIP2 into IP3 and DAG. IP3 is the main protein we'll be talking about here. As it follows the IP3 receptor channel, it will allow the influx of calcium to go into the cell. Overview of what we just went over for G protein-like receptors. As the ligand binds to receptor, it separates the G protein by removing GDP and adding GTP, which the alpha separates from the beta gamma, and from there it can either bind to adenylyl cyclase, which uses ATP to signal cyclic AMP, which activates PK, or it can bind to phospholipase C, which cleaves PIP2 into IP3 and DAG, which then, as IP3 goes through its channel, allows the influx of calcium into the cell.